Everyone, please, you can take a seat. Good afternoon and welcome to this round table titled Harnessing the Power of Medicinal Chemistry to Develop New Therapies, Inspiring Success Stories. Together with Marco, we discussed a lot about the topic of this round table. Maybe you remember that last year we had the COVID as a topic, but we said today we want to do, this year we want to do something different. And so I don't know what you are expecting by looking at the title of this round table. I'm trying to explain what was our idea behind this. And so we have already heard 2 times. The school has already started, but I heard 2 times how difficult, how exceeding long, costly. I would say there are some even adjective related to the drug discovery, like insane, okay? So today this was mentioned 2 times in 2 different presentations. The drug discovery has changed, and now it's something which is exceedingly difficult, okay? But when you start a career in medicinal chemistry, like you are doing, like we did, you have a dream. You want to bring a medicine, a drug, to the people in need, okay? So today I want to look at the problem from a different perspective, from the other way around, okay? Because my feeling is that you need, as a young scientist, motivation. Motivation is so important for your career. And so with Marco, we decided to tell you some inspiring stories of people who made it, despite the mission is impossible, because it's almost impossible. But today we will listen to successful stories. So this is the topics, this is the contest today. So I don't want to spend too much time, and the round table is organized around the presentation of the speakers, and then we will have some questions at the end, some questions from you, from the audience, for the people for who this round table was tailored, okay? So let's start. So the first speaker is Dr. Monica Binaschi. Monica Binaschi, she's a biologist. She made a PhD in molecular medicine. She's Italian, of course, as maybe you can guess from the name of Monica Binaschi. Yes, because I want even to stress this. And she was working in one of the most famous institutions in oncology, Istituto Nazionale Tumori, it's called. And then she moved to Menarini. Menarini is a big pharma. It was Italian. At the beginning, it was an Italian pharma, but now it's an international one, and it's quite big. And she's there since almost 25 years. And she will tell us a very successful story and a very recent one. So the title of her presentation is From the Discovery to the Approval of Elacestrant, the first oral SERD, you will learn what is a SERD in a while, in breast cancer. Please, Monica.
So thank you very much, Professor Bolognesi. It's a real pleasure to be here with all of you, students, to drive you through what's happened during these many years from the discovery to the very recent approval in January this year of elacestrant. Now is for the benefit of patients called Orserdu. So to drive you through all the data that has been generated, the excitement that really was part of the story of elacestrant, I will go through some information regarding the breast cancer and through main turning point for elacestrant through discovery, preclinical data, and the clinical efficacy that was then submitted to the FDA. So first of all, what is breast cancer? Breast cancer is a very heterogeneous disease. It's characterized by different subtypes. We have, let me check, where is the pointer? Just a moment. No? Okay, can you see it? Yeah. Okay, so we have hormone-positive breast cancer, which is characterized by two main biomarkers, which is estrogen receptor and progesterone receptor. We have HER2 breast cancer, which is characterized by the overexpression of HER2 protein, and we have what is called the triple negative breast cancer because it doesn't express any kind of biomarkers. Each of these subtypes is really treated in a very different way with a particular treatment algorithm. So I will focus on the endocrine positive breast cancer characterized by estrogen receptor and progesterone receptor positive breast cancer. The main treatment for this disease is the endocrine therapy that might be aromatase inhibitor, might be fulvestrant, many different kind of drugs that at the end, anyway, for the majority of patients, develop the resistance, there is disease progression, and then when you have an advanced breast cancer disease, the prognosis is really poor. So, as mentioned before, the main drug that are used to treat patients with HR-positive breast cancer are molecules that target this pathway, the pathway of estrogen receptor. Why is this so important? Because the estrogen receptor, as you might know, is a nuclear receptor that translocates to the nucleus when estradiol binds to the estrogen receptor. It dimerizes, sitting on the DNA and inducing the transcription on many different genes that are really important for the proliferation of breast cancer. There are different therapeutic strategies, what is called SERM, which is a modulator of estrogen receptor that binds to the same region of where estradiol binds. It sits again to the DNA, but blocking the transcription. Another strategy is SERT, means that disrupt sending the receptor to the proteasoma, is able to degrade the protein, so there is no more estrogen receptor sitting on the DNA. So I suppose during 2015, a very famous group at Memorial Sloan Kettering realized that after a few months or years of hormonal therapy, some mutation emerged in the estrogen receptor. So these mutations are really very rare in the primary tumor, but then it became really the reason for resistance to the endocrine therapy, and the majority of the patients, I would say 40 to 50%, according to different databases you can use, really developed resistance mediated by SR1 mutations. Again, this group at Memorial Sloan Kettering identified that the hotspot mutations are sitting in the ligand binding domain of estrogen. And what it drives this? Drives two main things. First of all, that the standard SERM are not able to bind anymore to the binding site, so rendering making the therapy less effective. But very importantly, now the estrogen is able to sit on the DNA because they are activating mutation regardless the presence of estrogen. So this was one of the main impressive discovery that came in the same moment when elacestrant was initially developed for vasomotor symptoms, so to reduce the hot flashes that in postmenopausal women you can have. And so based on this discovery and based also on really a great group at Duke University, led by Donald McDonald, they tried to understand the real role of elacestrant in the mechanism of action. And they realized that elacestrant is able to bind the wild-type estrogen receptor as well as the mutated estrogen receptor with a very peculiar mechanism of action, binding to this residue and inducing antagonism, and also to these other residues inducing degradation. So Radius, that was the originator also of the molecule, started to study this molecule exploiting this new information. And indeed, they demonstrated, using two different cell lines, estrogen receptor positive, that indeed elacestrant, here is called RAD 1901, is able to degrade the estrogen receptor and also to act, and you can see here in this diagram, as an antagonist, because it reduced the progesterone receptor whose expression is mediated by the binding of estrogen on the DNA. Last but not least, also elacestrant, more importantly, was tested in patients-derived xenograft. This is the most important model to be used in oncology to test molecule because it keeps the heterogeneity that you see in human tumor, and so you can test and translate the activity to human. Very impressively, indeed, elacestrant induces in all the models stable disease, as you can see here, for the majority of the model, in SR1 wild type, but also in different mutated models. In red, and it's in green, in red you can see the activity of fulvestrant. Fulvestrant is the standard of care. It is a degrader also, but is injected intramuscular. So here we saw the first oral SERD that can be administered into patients at home. And I want to stress one important part that is really fundamental when you test a drug for oncology. Why so many drugs fails? Because the majority of the time you test drug at doses that are not clinically relevant. So that's why at the end you can see beautiful tumor regression that is never translated into human. These models were done at clinical relevant dose, really clinical, both for fulvestrant and for elacestrant, and indeed was predictive of a good activity also into patients. So based on the preclinical data, the phase 1 study, dose escalation and cohort expansion, was started, and the activity was really impressive for a phase 1. So we observed an overall response rate of 33% in breast cancer patients heavily pretreated, and in particular this great activity was observed in patients resistant, harboring the SR1 mutations. Also, a very interesting finding was that using ctDNA, the circulating tumor DNA, we also were able to demonstrate that patients who were going to respond also had a decrease after the first cycle of the mutant allele fraction, so a pharmacodynamic activity associated with the inpatient's activity. So the results were considered really promising, and so we started the phase 3 pivotal trial. The trial was called Emerald, and the patients were mainly patients that progressed after a first or second line of treatment. All the patients had to be administered already with CDK4/6, which is the first line of treatment for estrogen receptor positive breast cancer associated with hormonal therapy. One important thing that I think was a really an highlight really important is that the patients were also stratified according to the SR1 mutation. So the statistical power of the clinical trial was really designed to demonstrate also activity in subpopulation. So the patients were divided to receive, it was of course a blind study, elacestrant or standard of care that could be fulvestrant, anastrozole, letrozole, or any other aromatase inhibitor. The primary endpoint of the phase 3 was the progression-free survival in all patients or in the subpopulation. These are the results. The results in the all patients, as you can see, elacestrant shows statistically significant PFS versus the standard of care in the whole population. More impressively, in the subpopulation associated with and harboring the SR1 mutation. In addition, we also analyzed the data according to the sensitivity to hormonal therapy. So patients that usually stay on treatment on the previous CDK4/6 are considering hormonal sensitive. And indeed, here we achieve in the overall population a PFS also even more statistically significant, but this was really, if you think that is a monotherapy, really impressive, a PFS of 8.61 versus the standard of care, which was almost 2. So, as you can imagine, when we saw the results, it was December. It was a very exciting moment because we realized that the patients really had an impressive benefit. And as you can also imagine, it took a village then to submit the data. Chemist, of course, CMC part, regulatory part, preclinical, toxicology, clinical, statistical, and really we work day and night to submit in very few months all the dossier that is needed for FDA and EMA to evaluate the activity of your drug. What else I can say? It was really, FDA granted us the priority review, is a channel that FDA use when see that the drug might really change the algorithm of treatment and is granted in general to molecule that fulfill an high medical need. And usually FDA takes 6 months to give you or not, of course, according to what they see, the result, the approval, sorry. And indeed, they were the most rewarding 6 months of my life because really, at the end, we arrived to have patients that can benefit from our drug. So, thank you very much. And,
Thank you, Monica, for this amazing story, of course, and I'm feeling particularly touched also as a woman because we know how spread is the breast cancer. And so thank you very much for all your effort and for giving the patients further opportunities. So thank you very much. And so let's move to the next speaker. Next speaker is Professor Vincenzo Summa. Vincenzo Summa is Italian. Again, maybe you can guess by the name. Then I will make a guess for you, Tony, if they understand where you are from. Okay, Vincenzo Summa is Italian, but he's a medicinal chemist who is internationally renowned. And he has a very interesting CV that I would like to bring to your attention because, okay, he graduated in chemistry, then he got his PhD in Germany, always in chemistry, but then he joined a Merck Research Laboratory in Italy in 1996 as senior research chemist. And then it took on position of increasing responsibility, including a director of the medicinal chemistry department. And, but then, this is not quite common, at least in Italy, he made an impressive transition because he came back to academia and now he's a full professor of medicinal chemistry at the University of Naples. The title of his talk today is The Discovery of Raltegravir from Hit to the First-in-Class HIV integrase inhibitors Approved, Failures and Success. So, completely fitting our roundtable.
Thanks, Maria Laura, for the kind introduction. So my talk will focus on really the history of this, the discovery of this compound. It is the first-in-class HIV integrase inhibitor approved. I want to start with some facts. So we have to go back to the 1996, 1995, where no cure, no vaccine were available. There were some HIV reverse transcriptase inhibitor approved and the first HCV protease approved. And with this combination, these three drugs, we started the triple combination therapy that was really the turning point for the HIV patients. Integrase had, Merck had already both reverse transcriptase and protease inhibitor approved. And so their choice was really to focus on HIV integrase as new mechanism of action. Today, we are not very far away where we were, so there is still no cure and no vaccine. The therapy is much better. We have single pill that is very efficacious, a combination of different drugs, very efficacious. So we are still looking for a cure of the patient. So we have to think that HIV is a treatable disease, but we cannot cure the patient. It is all for the rest of the life. What is the situation? HIV is still a problem. So we have 41 million people infected, almost 1 million deaths every year, and every year we get 1.5 million new infections. If we look here, so North America and US, we have quite a big number of new infections and people that are living with HIV. Although we are not talking anymore about HIV, it's still a really big problem. Just to understand what does the HIV integrase. So we knew that the virus has to go in contact with our cells. There is the fusion, the release of genetic material. The reverse transcriptase convert the RNA into the DNA. These little boxes are the HIV integrase that are bound to the viral DNA, enter into the nucleus, and the integrase integrate the viral DNA with our genome. So this is the step of the integration. For this reason, we cannot cure the HIV patient at the moment. We do another zoom. just to understand the mechanism. So the integration is a multi-step complex process. The first one is the independent processing. So we chop two nucleosides at the end of the viral DNA, enter into the nucleus, this complex, and this step is a concerted, is the real integration, is the strand transfer integration, in which we have the cleavage of the host DNA and the link with the viral DNA. We do another zoom just to understand the molecular mechanism. So how does work the integrase? We have two magnesium that are the engine of these enzymes. So they are able to catalyze the reaction. They activate the 3' OH on the sugar, make the nucleophilic attack on the phosphate of the host DNA, form this pentavalent intermediate. There is the strand transfer, and now the viral DNA is really linked, covalently linked with our DNA. There are many enzymes that are working with the same engine. So they have always magnesium in the active site, but they do different reactions. One of them is the polymerase of HCV. When I joined Merck, I didn't work on HIV, but I was working on HCV, and was working on the polymerase. This was my target at the time. So the polymerase works exactly the same. So the magnesium activates this OH. Instead to have the joining with the viral DNA, with the release of the pyrophosphate of the triphosphate. So we have the incorporation of the nucleobase. This is a strand elongation. So it's not integration. There is translocation. The reaction of polymerization continues in very quick manner. So Merck clearly had really big sample collection, so what we call the collection of all Merck compounds, million of compounds, 2.5 million compounds. And so we ran for both programs the HTS against the two targets. And this is interesting because at the beginning we really didn't understand anything about the mechanism of action. So the integrase team in the US found the diketoacid, this molecule here, as a bona fide inhibitor of the strand transfer process. And from our side, we identified the diketoacid that were bona fide inhibitor of the HCV polymerase. Took quite a lot of time to understand how these molecules are able to work. And the reality, we discovered that both molecules have in common this diketoacid moiety that are able to chelate the magnesium in the active site. So basically, we block the engine of these enzymes. And the green part, instead, is responsible for the selectivity. So changing the green part, you change the selectivity between the two enzymes. So in fact, the molecules on the left are selective for integrase, on the right are selective for the HCV polymerase. Unfortunately, the diketoacid are very pure drug-like molecules. So we immediately learned that the PK properties are very poor, are not chemically stable or biologically stable, and furthermore, they are able to alkylate irreversible our protein. So this is something that we don't want. So we worked very hard to find an alternative scaffold, and in my shop, we did a second HTS campaign, and we come up with the meconic acid derivative. You can recognize in red the part that is always responsible to chelate the magnesium in the active site. Also for this series, we were able to really bring the potency very down, but we didn't solve some of the issue of the diketoacid. So these are chemically unstable, and so when they lose the carboxylic acid, they lose the activity. We designed this nice scaffold. It's very fully decorated, so it's the hydroxypyrimidine carboxylic acid. So we solve all the problems of the diketoacid, apart the PK. PK properties were really poor. And so what we discovered was that this was due to the fact that we have this carboxylic acid, but also this OH is very acid. So it's bis acid. And this led really the poor PK. So we try to modify, you use all the bioisosteres of the carboxylic acid, nothing worked, but we did one thing that was, in a sense, without great hope. And we made an amide and an ester, and the amide turned out to be very active against HIV integrase, and completely inactive on HCV polymerase. So for serendipity, looking for the carboxylic acid replacement, we found very potent compound HIV integrase. So we asked our colleagues if they were interested in working on this series. They said, no, we are busy with our molecule, so if you want to join, join. I did, and this was really my turning point in the program. And I changed the program from the polymerase to the integrase. So just very quickly, so what we are looking here are the enzymatic assays that are called quick-in. They measure the inhibition of the strand transfer. And then the spread assay is an antiviral assay in which we measure the inhibition, 95% of the inhibition of the viral replication. So we abolish completely the replication. Going back to the therapeutic dose, what we want to have is a compound that in plasma, circulating in our plasma, is as a concentration above the IC95 of the spread assay in presence of high serum concentration. And the number is 100 nanomolar, because at the time that was the concentration of the approved drug. So we use two different concentrations of serum, low serum, high serum. So low serum reflects the cell-penetrating property of the compound, and the high serum condition that is much more physiological reflects also the effect of the protein binding on the compound, on the potency. The other thing that is very important, if we have a compound that is completely bound to the plasma, the metabolism of this compound is very, very low. So there is a balance between clearance and activity. So we need to have a real balance. This was our lead. You can imagine that we work around all the molecules. So we did a great screening on the amide. The parafluorobenzamide was the best. One thing that we learned very quickly, that the position to the pyrimidine, we can put anything we want. And reality is that we can clearly take off completely the substitution in the position 2, and we maintain the activity, same level of activity in the enzymatic assay. Still, these compounds are really poor active in the cell-based assay. What we learned was that if we put in a basic amine, we can gain potency in the spread assay, so antiviral assay in low serum condition here. So twofold shift between the enzymatic assay and the spread assay. But when we go to the high serum concentration, we lose completely the activity, because this is completely bound, so 99%. On the contrary, this other compound is the opposite. We have big shift between the enzymatic assay, the antiviral assay in low condition, and we don't have shift in the high serum condition, because we have 10% free in, on the protein binding properties of this compound. One thing that I forget is that the compound with higher protein binding has much better PK. So we decided to focus on the compound with better PK more than the compound with the best potency, because it's much easier to improve the potency than the PK. And so these are the two strategies. The first one, we removed completely the aromatic ring. We come up with a simple achiral compound that finally achieved the potency that we were looking for, the famous 100 nanomolar in low serum concentration, low serum condition in the antiviral assay. We developed the compound. We made the crystalline ACL salt, because we went in the phase of trying to develop this compound, very clean and different assays. The PK's properties are very good, especially the DOG is 100 nanomolar oral bioavailability, very high exposure. And so the DOG is the reference species to predict the human PK. So every time that you try to develop a compound, you need to find the species that can predict the human PK. So in this case was the DOG, and so we predict to have a BID drug that was good enough. Unfortunately, the compound was positive in the ancillary pharmacology test, and particularly in the CNS mouse. A very high concentration. We try to say that this doesn't make any sense because it's too high. The concentration is 1 gram per 1 gram of compound per gram of animal. Was unbelievable high. I say, no way, that you cannot develop the compound, so we discontinue the compound. So we get really depressed about this. The other thing that clearly we never stopped, so what we learned was that we could make the N-methylpyrimidone instead of the hydroxypyrimidine analog, and the effect is quite clear. So the protein binding is drastically reduced, it's much more polar, and the oral viability is increased. So we focus our attention on the N-methylpyrimidone, and modifying the substituent in the position 2, you can imagine making the N-methyl chemically is much more difficult, and the morpholine is a chiral compound, another additional problem in the compound. But nevertheless, we made grams of this chiral compound, HCl salt again, very potent, less than 100 nanomolar in antiviral assay, in high serum condition, very soluble. Again, PK, very nice in 3 species, especially in dog. And in this case, we had the human PK prediction to be once a day. We were particularly happy about the results, and we were more happy when we see the compound clean the CNS mouse that was the previous killer. But the long story of the development of compound just was another stop because we found the compound very positive in Ames assay, that's the gene toxicity assay. Again, an incredible concentration, 3 grams per plate. So 3 grams of compound in a dish, extremely high. We made the case with the FDA, they say, no way, is again, this compound is dead. And so we discontinued the compound, and we put the Ames assay in the early phase of the screening cascade to evaluate the compounds. For the CNS mouse, the previous compound was the only one that was positive. For the Ames test, we found other compounds that were positive. So we were very depressed, but our depression didn't finish there, because our colleague in West Point, clearly they realized simultaneously with us that the keto are not good. They were able to replace the carboxylic acid with this keto heterocycle. They made a very nice constraint of this keto heterocycle, creating this naphthyridine hydroxyketone, very complicated to make and very insoluble. When we come up with our benzylamide, was obvious to make the naphthyridine hydroxycarboxamide. And this was the new lead for them. And in fact, as in the case of the pyrimidine, here we can put anything we want. And so they made a lot of different substituents, but 2 compounds are very important. This one that contained the oxalamide here, that was very potent, so was potent and with nice PK, not potent enough to be developed for man, but was potent enough in the RSV, so is the equivalent of HIV for the rhesus. And so we did the first proof of concept that an HIV integrase inhibitor is able to abolish the replication in animals, so in a real system. So this, because of the importance of this drug, this finding was published in Science. The second compound is this sultan ring in the position, achieved the 100 nanomolar potency, and this compound went straight to the development. So was clean in the ancillary pharmacology test, was clean in the gene toxicity assay, was clean in the 14-week safety. So we went in phase 1B, because at this point was really, we want to see if this compound is efficacious in men. So we went in the naive patients, and this is the baseline, this is the placebo, this is the viral load, didn't have any change. And the people treated with the 810, that is the previous compound, after 10 days, they got almost 3 log, 2.7 log reduction in the viral load. Very efficacious, very incredible achievement. This is a monotherapy. So we can do the monotherapy only if the mechanism that we are investigating is completely new. Otherwise, you have to combine with the other drugs. But as I said, we were quite happy. We were in phase 2, and instead, in the long-term safety, 9 months safety in dogs, the compound was toxic. This toxicity, we proved that was species-specific. For example, neither in rat or rhesus we have this effect, but nevertheless, ESMEC decided, no way, although we know that what happened in the man is always a risk, so we stop again the compound. So at this point, the morale of the team was below 0, because we lose several compounds in the preclinical and one in phase 2. So, but we never stopped to work. So we continued to work on our N-methylpyrimidone, and in particular on the carboxamide, and you can recognize the oxalamide, that is a moiety that we took from the West Point series, very potent, at 74 nanomolar. We made a co-strain of oxalamide, and we got even more potent compound, MK709, MK518. So the question was which one of the two compounds we want to develop. The answer came out, we never talk about the resistance. Clearly, in the real field, so in the HIV patient, they are not resistant to HIV integrase because there was no drug. But what we did during all the program was really to select mutants to see which one has the highest genetic barrier to be developed. And if we compare, this is the naphthalene compound that failed in phase 2. This is the compound with the oxalamide, our compound 11. This is MK518, that is the compound with the oxadiazole. And this is a compound from a competitor. So you always monitoring also the competitor. MK518 was much better, so we took 518 and we went straight to development. So clean against all these different enzymes. The PK was very nice in dog, was clean in ancillary pharmacology and toxicity assay, is not cytochrome P450 metabolized by cytochrome P450, but just glucuronidation. This is really a plus, because you reduce the possibility to have drug-drug interaction. So with this data, we went in naive patient and in salvage therapy. This is more important, the salvage therapy. So naive patients are people that never saw any drug. So we did first the healthy volunteer in monotherapy, and you can see all doses. We have 2 log reduction for all patients. With this data, we went on with the clinical trial. This is our phase 2. These are 96 weeks. You can see here for the first 48 weeks, we bring on all the doses. And at week 48, we decide the final dose, that is 400 milligram BID. And you can see that all patients we don't have any relapse, any selection of mutants, so the therapy is very efficacious, and so this means that it's a very, very, very active drug. More important, this is the salvage therapy. So this is the optimized background therapy for the patient that they are resistant to all drugs. The physician select, so make the screening of the virus, find all the mutation, and make a combination of 3, 4, 5 drugs. And each patient has a different therapy. And so it's really personal therapy. And you can see that only 20% of the patient can respond to this treatment. What we did was to put on top of the optimized background therapy the raltegravir. And so you can see that almost 100% of the patient really rescued completely. So we were able to really salvage this patient. And this was the real fast track approval. So this data led us to get the approval in 2 years and 9 months from the approval of the preclinical candidate. So we got the approval with this, and then first-line therapy later on. So the American Chemical Society recognized the team that worked on these are the West Point team and the IBM team as a heroes of chemistry in 2000, I don't remember, 7. And more importantly, the iCenter got the Prix Galien, US and EU for the best pharmaceutical agent in 2008. this is a recognition of the impact of this drug made on in the HIV field. So the teams are really spread around the world. West Point, Rome, chemistry, Rowe and Paris. Rowe and Odesson, process chemistry that always accompanied, were really good friends in all the development of the program, and Paris and Bagno, toxicology. So this is the list of the people. This is a part of this, mainly is a basic research team, and just because we just with Maria Laura, so which was my dream, was really make a drug, and I hope that you have the same dream, but the question is why we are making drug. I like to really get this because, you know, I thought that this phrase from George Merker is 100% true. If you are able to make a good drug for the people, clearly you have an impact for the society, but then you get also the profit. If you try to do the opposite, make the profit with the drug, you are on the wrong path. And having said that, I want to thank you for your attention. I think I went long.
Thank you very much, Vincenzo, especially for the ethical part of your talk, because I want to stress what Vincenzo said. Okay, you are so young, you were not even born when HIV was a major threat. But maybe you have seen the movie Philadelphia, don't know if you, but in any case, for us it was so scary, such a big threat. And so, but today we shouldn't forget all the people, all the population which is affected by HIV in the poorest area of the planet. So we should even consider how to get the medicine accessible to those people, even to the children, no, or even the malnourished patient. We have HIV together with other infections. So I believe that we should still consider this kind of drugs. And again, thank you even for this remark. Okay, so now let's move to the next speaker. The next speaker is Matilda Tilly Bingham. I'm truly honored to have Tilly with us today. Unfortunately, Tilly couldn't make it in person, so she will be connected from UK. And Tilly, oh yes, maybe if you want to switch on your camera, I'm going to...
I have my camera on. How do I make it show? Yeah.
Okay, yes, we can see you, okay. I was telling that I truly thank you. I'm so glad, so honored that you are participating, even virtually. So I would like to say a few words on your career. Tilly Bingham is a drug discoverer with almost 20 years experience of working in the pharmaceutical R&D sector, both large pharma, organ sharing plow, MSD, and biotech. And she's currently VP of Science at Concept Life Sciences, overseeing multiple programs in Hit-to-Lead and lead optimization. I want also to emphasize that Tilly also works a lot for the community because she is involved in the Royal Society of Chemistry, and also she's also elected a member of the Executive Committee of the European Federation of Medicinal Chemistry. So now the floor is yours, Tilly. And the title of her speech is, as you can read, The Rough Paradox, Not All Kinase Inhibitors Are Equal. Very intriguing to me, and also to all the audience, I guess.
Right, is that gone full screen?
Yeah.
Yeah, great. So I hope I have done a presentation here that fits the remit of the meeting. So to begin with, I thought I would just give you a few thoughts on a medicinal chemist's career path as it is in industry, because my background is a little bit different from some of the other speakers. That I'm from a completely industry background. I didn't start out that way. I started out, as many of you probably in the room are, as an undergraduate PhD student and then as a postdoc in academia. And I really took advantage of what this career gives you in terms of the ability to travel. So I worked in London, in France, and in the States in my early career. I then, in 2000, moved to Scotland and started my career in industry, working in a pharmaceutical company. And the first 10 years of my career, I worked as a medicinal chemist project leader in the area of neuroscience. And I worked on many, many targets, a lot of GPCR for peptide hormones and reuptake transporters. I then moved to a different type of career option in industry, which is a biotech company. There I was head of medicinal chemistry and then head of R&D. And there I worked on oncology and immunology programs. So a complete change, which you can do when you're a medicinal chemist, change therapeutic area. But that's the therapeutic area, the two therapeutic areas I've really remained in. And then back in 2018, I moved to the third option for somebody in medicinal chemistry, which is to a contract research organisation. So here we're still doing the medicinal chemistry and the projects and programs, but we're doing them for various clients, still in the areas of immunology, oncology, and neuroscience. What choices you make when you move to go into industry is that you choose to become anonymous. That suits me. I'm quite happy. I don't do many talks. I'm quite happy to sit in the background. But as a consequence, it's often quite difficult to present talks because the programs that are successful are the programs that the companies don't want you to talk about and give too much description on. So actually, although I've worked on a couple of launch drugs, the CGRP antagonist program I worked on at Merck was actually sold to Allergan, then to AbbVie, and was approved in 2021. And also the BTK inhibitor program that I worked on at Redex just got approved this January. But I'm not able to talk to you about those today. What I'm going to talk to you about is a different program, is a RAF inhibitor program that was sold to Jazz Pharmaceuticals from Redex. And because of the limitations, there's certain limitations on how much information I'm able to give, but I think there's enough to make it a nice story. So what is BRAF? So BRAF is a kinase that's involved in many cancers, actually. And the RAF kinases themselves are key regulators of cell proliferation and survival, and they control signaling through the MAPK pathway. And there are three of these kinases, ARAF, BRAF, and CRAF. And the one that's most prevalently mutated in cancer, in fact, in 8% of all cancers, is BRAF. And in two particular cancers, melanoma and papillary thyroid cancers, it's extremely prevalent. And the most common BRAF mutation that occurs is at position 600, where a valine is substituted by a glutamic acid. So for those of you who might need a memory jog about kinases, kinases are involved in phosphorylation. So they take a molecule of ATP and they transfer a phosphate group to the substrate, which is not normally another protein. And in the case of BRAF, it's another kinase called MEK, which then goes on to phosphorylate a further kinase in the MAPK pathway, ERK, and that leads to proliferation, cell growth, and survival. So the inhibition of BRAF is actually, at the point that we started this program, was a clinically validated approach. So there were two known drugs that had just been approved in 2011 and 2013 for the treatment of BRAF V600E metastatic melanoma. And the initial responses were very good, in fact, astounding. But the resistance to treatment developed over time, 6 to 12 months. And as you can imagine, this could be very upsetting for patients to have seen almost, you know, a miraculous recovery followed by the cancer returning. There was a further problem in that they were not effective in RAS mutant cancers. And in fact, they actually accelerated tumor growth in RAS mutant cancers, such as squamous cell carcinomas. So given squamous cell carcinomas are slow growing, that was considered an acceptable risk, but obviously one which you would want to avoid. So how did this mutation work? How did it accelerate the signaling in the pathway and lead to upregulated phospho-ERK signaling? Well, that was proven. It did indeed work via upregulating ERK signaling. But if you look at where the mutation is, can you see my cursor in the room?
Yes.
Yes, okay. So if you look at where the, oh, it's disappeared now. Yeah, if you look at where the mutation is, it's actually not in the ATB binding site, it's at the dimer interface. And in fact, the RAF kinases are shown to act as heterodimers and homodimers. And what was happening in the BRAF V600E is that the mutation through a network of bonds was disrupting the dimer interface, leading it to being proficient to signal as a monomer, and that was leading to the accelerated pathway signaling and thus cancer in this mutated population. And that, in a cell assay, looked like this. So there was actually a cell model which was able to replicate what was happening in the clinic, such that we could begin a program to look to model this in vitro. So if you look at the graph on the left in green, you can see in a RAF V600E mutant cell line, increasing concentrations of the compound vemurafenib, which is one of those I just told you had been brought to market in 2011, you can see inhibition of phospho-ERK signaling. If now we're moving to a RAS mutant cell line with wild-type BRAF, the opposite is happening. So you are seeing an increase in phospho-ERK signaling, and only at very high concentrations do you start to see a decrease in that signaling. So what's happening? Well, when I showed you the image before, I didn't show you quite everything. And actually what happens when these agents bind vemurafenib is that they only occupy one protomer of the BRAF dimer or heterodimer. So for those in the audience, I'm going to give you a few seconds to take a look at these two graphs and think in your minds what your hypotheses might be for why is this happening. Why is that molecule decreasing phospho-ERK, increasing it in the other? And that was the challenge for our program at the time. Well, the answer is the difference between the inhibition of the monomer and the inhibition of the dimer. So in the case of the RAF mutant cell lines in cancers, you have a monomer, it's inhibited by vemurafenib, and you see a simple trace. In the RAS mutant cell lines, actually what you see is activation of the other protemer of the dimer in the initial phases at partial occupation. And it's only when you're at very high concentrations of vemurafenib, because its affinity is reduced at the second protemer of the RAF, that it begins to be occupied and you start to see inhibition. And that's why, as you're at low concentrations, you start to see monooccupation, and that reflects activation, and it's only at high concentrations that it starts to come back down. So thus, that was the state of knowledge at the point that we began our program, and we considered what are the strategies that we could apply here. Well, there are two very different strategies, and actually we had programs within the team pursuing both of them. So the first one is to say, let's just find a compound that's so exquisitely selective for the monomer and BRAF V600E, and it will never bind to a dimer at all. And this would thus be a vemurafenib plus type compound. So it would have a very similar profile to vemurafenib without the side effects. And the second approach was to find a compound that inhibits all of the RAF wild type isoforms, and particularly CRAF, because it had been identified that in that dimer setup where you're activating the other part of the dimer, the most prevalent RAF isoform that was occurring through was CRAF. And that's the approach that I'm going to go on to tell you a little bit more about today, the second approach. Here you could envisage that this agent could be active not just for the V600E mutant cases, but also for RAS mutant tumours. So that all sounds very nice. Why hadn't people done that in the first place? There must be something that's challenging there that had been why the previous programs hadn't taken that approach. Well, there is a challenge. So ATP, as I'm sure many of you know, is present in cells in millimolar concentrations. And what that means in the case of the RAF signaling pathway is that you can see differences in activity when you move from your kinase assays into cell-based assays due to a drop-off competition with ATP. And so we knew that the strategy that we were selecting was going to be a challenge, because if you look here on the left, you can see the KM-ATP apparent for the mutant is 65 micromolar, whereas the KM-ATP apparent for BRAF and CRAF is in the 3 to 5 micromolar region. So this means it's going to be a lot easier to compete with ATP at the BRAF V600E kinase than it is at the BRAF wild type and the CRAF wild type. And in fact, you can see this for vemurafenib. If you look here in your typical kinase assay that you would get from, I don't know, DiscoverX, whoever you send your kinase profiling to, you could have a nice warm feeling if you looked at the Ki, because you would see nice 4 nanomolar for BRAF V600E, 2.6 nanomolar for wild type BRAF, and 3.3 for CRAF. But actually, if you then look up the same conditions, but with 1 millimolar ATP, and here you're going to mimic more what will be happening in the body, you can see the numbers are very different. So there still is a drop-off in the V600E cell line, but in the wild, almost to give near lack of efficacy in those cell lines. So we knew there was going to be a challenge in terms of ATP composition if we were going to pursue the wild-type proteins. So what was our stated game? We wanted a potent pan-kinase inhibitor, and we'd identified that activity at CRAF was key, and we knew that we would have this challenge with competing against the high ATP concentrations and higher KM-ATP apparent for CRAF. So we elected to pursue a strategy where we would target what's called a type 2 kinase inhibitor to focus on inhibiting the inactive form of the kinase, which would facilitate the goal of the program. And I'm not able to show you any of the structures, unfortunately, of how we did that, but the program was ultimately successful. And I can take you through some data from one of the examples that we've published on here. This was presented at the AACR meeting in 2017. And if you look over on the left, you can see that in HCT116, which is a RAS mutant G13D cell line, the REDX compound inhibits phospho-ERK. If you look at the 10 micromolar time point, you can see along the row at the equivalent 3-hour time point for vemurafenib, there's significant activation there of phospho-ERK at 3 hours relative to DMSO. And then if you look below at an example of an NRAS rather than a KRAS cell line, you can see that the same is true when you're comparing the REDX compound with vemurafenib, again, in that NRAS mutant cell line. And on the right-hand side, you can see the compound had excellent kinase selectivity and was potent against the 3 main isoforms, ARAF, CRAF, and BRAF, including BRAF V600E. And when we extended this to look at a broad range of RAS mutant cell lines, we saw that this observation could be replicated across the board in terms of anti-proliferative activity. And also, when this was then taken in vivo with an example where we knew we had good pharmacokinetic properties, we were able to see prolonged tumor growth inhibition, and we were able to correlate that with in-tumor pharmacodynamic response on phospho-ERK, and you can see that represented with the graph on the right. So where are they now with the program? Well, in a way, one of the things about being a medicinal chemist in industry is that once you've reached the end of your program and nominated your candidate, you hand it over to someone else, and then you have to watch in the news, you know, as much as everyone else as you're finding out what's happened to it. So really with this program now, I'm following it from a distance and I get to know as much as you. But the latest news that has been published was actually just last month, and Jazz Pharmaceuticals and REDX, their candidate from this work that we did, is entering clinical development. So that's excellent news. And yeah, that's my talk. Thank you very much for hosting me.
Thank you very much, Tilly, also for the final personal note, how you feel when you start a compound and you feel like the mother of that compound, and then you continue to follow the compound, even if it's in other hands. Yeah, I truly thank you for all the story. The paradox is very nice. I also, as you managed the paradox, and also, yes, thank you for this personal note at the end, and even at the introduction. Thank you. So now Last but not least is the turn of Tony Torrens. Tony is a long-standing friend of the Urbino School because he has been the delegate of the European Federation of Medicinal Chemistry in the scientific committee of the school. So he has participated to several editions and contributed to the program. Regarding his career, Tony graduated in pharmacy and was awarded a PhD from the University of Barcelona. He did a postdoctoral stay in Paris, and then he spent most of his career as head of the medicinal chemistry in Esteve. Esteve is a huge pharma in Spain. I know personally by reviewing many papers in ESMEC that they had programs in analgesia. This was the main focus of the company. But now, as we said, that in industry transition is quite common. Now, the present affiliation of Tony is working at ABAC Therapeutics in the antibiotics field. The title of his speech today refers to an interesting case study, and the title is a co-crystal combination for analgesia. Please.
Thank you, Maria Laura, for the nice introduction. Indeed, I'm a big fan of this school, summer school in Urbino. Thank you for the opportunity also to show this case study. However, to be the last one today is very tough because after the impressive stories from my colleagues, mine will be a little bit humble, but nevertheless, I would like to show you also the message that with a small innovation and pursuing a lot and working hard, you can also deliver compounds to the market. And this is what I'm going to explain today. As Maria Laura said, this was done at my former company, Esteve Pharmaceuticals in Barcelona, a company which has several compounds in phase 2 for analgesia. But the big success was this combination that we achieved by doing a co-crystal. So first of all, just a few words about the prevalence of pain in the US and worldwide. It's still huge, the number of people who suffer from pain, daily pain in the US, more than 25 million. Over 24 million American adults report a lot of pain. Most of them experience category 3 pain, but even 14 million category 4, that is real stressful, and this is a burden for all the society, mainly for the healthcare security of most of the countries. Also, the pain management, it's not easy. I can tell you that it's quite challenging because the analgesics that we have in clinics sometimes are not enough, sometimes are too heavy, and sometimes are not effective. So when you have moderate pain, you usually are prescribed with non-opioid analgesics. But as soon as you have a severe pain or mildly severe pain, you could end up with opioids. And as you well know, opioids have a lot of problems. I will show you later on. So this can be seen here. Going for mild pain and poor secondary effects, we have very few compounds. But as long as you go to the severe pain, you end up with strong opioids, morphine, oxycodone, and nowadays fentanyl. It's even worse, heroin. So what the scientific community usually try to do is to go and have more active compounds, and in the case of opioids, this has been a real drawback because there is no opioid, as far as we know, that is not promoting tolerance and promoting the liability of drug abuse and overdose, okay? On the other side, the compounds that are in that part, inside COX-2 inhibitors, paracetamol, acetaminophen, they have a good tolerability, but of course they cannot be used for severe pain. So there is this huge unmet need here that we need strong analgesics that do not promote any kind of liability. Because as you can see here, and this is quite dramatic, I must say, you can see here that in the year 2021, more than 80,000 people died in the US by overdosing. This number could seem not too big, but imagine the biggest calcium stadiums in Italy, San Siro, Olimpico de Roma, cannot allocate all these people. So these people do not fit on those big stadiums. So it's a drama that now is also all over the world, but also in Canada, mainly in British Columbia, with the appearance of fentanyl that is dramatic. A lot of people is dying also in Canada now. So we thought that probably to go for this unmet need here, probably a combination could be a solution. So we try to use weak opioids and COX-2 inhibitors or weak opioids and NSAIDs to try to decrease the dose of the opioids, and in this way to try to overcome the liabilities. Mainly tramadol is one of the less used or for abuse opioids, because it works with a different mechanism of action. It's not only a mu-opioid agonist, but also it is a serotonin reuptake inhibitor and norepinephrine reuptake inhibitor. And in high doses, it promotes the vomiting. So people do not abuse very much with tramadol because, of course, it's not pleasant when you are partying or when you are abusing to vomit. And so we thought about tramadol and to combine it with some other NSAID or COX-2 to get this tramadol from here to that part to get a more efficacy of tramadol or less dose to have the same effect. And what we did was look into the bibliography. There are some clinical evidences. I'm not going very much into detail, but there are publications where these are all fixed combinations, or fixed combination of the two compounds given at the same time. And there are several articles that show you that this kind of combination could enhance the efficacy and at the same time could reduce the dosage from those compounds. There are plenty of them. And of course, the scenario of the multimodal or multi-target therapy could be those three scenarios: 2 tablets, 2 drugs, when you given the compound separately. This has a lot of advantages and cons, of course. The advantage is that you can give the dose that you want and the periods that you like to do. Then we have the 1 tablet, 2 drugs, that this was our approach at the end. And this is what we were pursuing at Esteve very much. 1 tablet, 1 drug. So new compounds that have more than one mechanism of action, so dual targets, what we call multimodal analgesia. So there are compounds in phase 2 for Esteve from this type of compounds, dual ligands or even triple ligands. But the one who makes it to the market was this one that I'm going to explain today. Believe it or not, combinations are not just a trivial thing. There are blockbusters that are already a combination. You can see, for instance, at Bayer, luticasone and salmeterol. They made a lot of money in 2005. But as a professor said before, Professor Suma, the main goal here is not to make money, is to treat patients and to cure them. And so I think that this is something that needs to drive our efforts every time and everywhere. So we think on doing something a little bit different, not just mix a combination, mix 2 physical compounds, but to have some kind of bond or some kind of strong interaction between the 2 drugs. We thought about making salts of two drugs, to have them with an ionic link, ionic bond, or to have co-crystals when those compounds cannot form a salt because of their chemical structures. So we explore both. And we started in 2007 here, we started with this patent. You know that sometimes in the industry we used to patent and not to publish. So mainly I will show patents today. And the first salt we made was this tramadol S-naproxen salt. And I have to tell you that we got a very interesting salt that was the stereomeric resolution. And to be honest, even though the compound show interesting results in animal models, what was the best here was the method of the stereomeric resolution of tramadol, which is not easy. And we found by chance this methodology, naproxen, is very cheap. So we patented this later on as a method, industrial method for the stereomeric resolution of tramadol. But this was the first salt in 2007 with this API combination. And then we moved to others. As I told you, we thought that tramadol was really a very good drug, a very interesting drug that is not, people do not abuse with it. And so what we try to do is to enhance the efficacy of tramadol by any combination. Then if you look to these two structures, because telecoxib, you know, is a COX-2 inhibitor that do not have any cardiovascular side effect. It's in the market for many years as Celebrex. If you look at the structures, you can see that tramadol clearly is a tertiary amine, but, and that's why it could make the salt with hydrochloride acid or with naproxen or whatever. But if you look at telecoxib, it's a compound that cannot make any salt, at least in aqueous media. Of course, you can do the salt of this part, but not in aqueous media. And then thelecoxib is a very insoluble compound. And this is the main drawback for Celebrex. They need to micronize and to have a structure of 3 microns or 5 microns to get absorption in vivo. It's a really a rock. You cannot dissolve the thelecoxib easily. In water is 4.3 milligrams liter at 25 degrees. On the other hand, tramadol is extremely soluble, highly soluble, tramadol hydrochloride in this case. But as I said, it's a weak opioid and a Schedule 4 substance, because even though this is the less dangerous, it's still dangerous. Then what happens with this? You cannot make a salt with those two compounds. Then we thought about doing a co-crystal. And believe it or not, because now you know the end of the story, and you know that I will explain the success of this, when we started with this, there was no API co-crystal described. So we looked at the bibliography extensively, and we found that we were the only ones. And that is always good, but also can be bad, because probably it's a bad idea if nobody else is doing this before. But at that moment, we were the first. Later on, I'll explain you later, a big company was the first in delivering API co-crystal to the market. But at that time, well, we were pioneering and started to do a lot of tests, of course, several solvents, grinding, all the crystallography or the crystal engineering stuff. And to make a long story short, we get a nice co-crystal with grinding by isopropanol at the end. That was, after all, in CMC, was perfectly scaled up. Just to not to go very much into details, but to see that here you have the 3D structure of the co-crystal, and the two main symptoms I just depicted here in 2D. Hopefully I should have this virtual reality with me to show you the 3D structure, because it's really nice. But here I can show you the two symptoms, the main symptoms. The driving force for the co-crystal is the hydrogen bonding from the NH of tramadol with the chlorine anion, and also from the side of thelecoxib, the NH of the sulfonamide. On the other side, there is this other one, the oxygen of the sulfonamide can also display a hydrogen bond with the hydroxyl group of tramadol. So, and this forms a perfect co-crystal that we scale up, and we go for development with it. This co-crystal was obtained in 2008, And from the very beginning, it showed advantages in physicochemical properties, mainly a better dissolution rate for celecoxib. That was a challenge. Also, the PK in animals of the co-crystal had better profile than the mixture, the fixed combination of the two drugs. We could reduce the regular dose of tramadol in 12%. We end up in 88 milligrams instead of 100 milligrams. And the preclinical research and CMC synthesis were faster than usual. CMC was really, really good in our sister company, Steve Chemistry, Steve Chimica. They did a great job, and they were able to synthesize even tons of the co-crystal. As expected, no issues in toxicology studies, but I'm just going to show you results in human, because when you have results in human, are the most important ones. So the phase 1 clinical trials were straightforward, and we got positive phase 2 studies in 2013. So it was 5 years only from the discovery to the phase 2, but you will see that phase 3 lasts much longer. And these are the pharmacokinetic in humans. Very interestingly, you can see here that we have Seglentis, which is the trade name of the initial name of the compound, Esteve 58425, sorry, and Seglentis the trade name. This is Seglentis, this is celecoxib, but you can see here that by any reason, and we already knew that, because tramadol and diclofenac were already described, but not tramadol and celecoxib, because we knew that this fixed combination had a very bad pharmacokinetics. And so this could be enhanced by the co-crystal, and we almost get the same level of results of celecoxib itself. So that was really, really good, and this proved that in a way the co-crystal helped the pharmacokinetic of the compound and the release at the same time of the two compounds. And the mechanism of action is quite complex. It's well known, but quite complex, because it's 4 different mechanisms of action. You have the cyclooxygenase 2 from the COX-2, inhibition of this cyclooxygenase in the prostaglandin cascade of inflammatory pathway, the arachidonic acid pathway, exactly. And then tramadol is quite complex, because the mu-opioid receptor partial agonism is not due to the drug itself, but a metabolite of the drug. So tramadol has a methoxy group, and this methoxy group is cleaved to the phenol group, and this phenol group, only one enantiomer, is very, very active. And someone tried to deliver this metabolite to the market, but it was not allowed, because as soon as you get this metabolite, you abuse with tramadol, because you don't have the other components. The other components that are serotonin transporter inhibition and norepinephrine transporter inhibition, and also a slight effect on the 5-HT3 central regulatory vomiting mechanism. As soon as you avoid this and you only prepare the pure enantiomer phenol, that is the opioid receptor partial agonist, you end up with problems the same as probably morphine. Okay. Then with these four different mechanisms of action, the co-crystal has a very balanced mechanism and promotes no side effects at all. Okay. So then the phase 2 was positive. As I said, this is a script report on 2013. Here it says that Esteve was looking for a partner to go to phase 3, but finally we were so excited with the result that finally we did it by ourselves. So it was a case that we invest all the money by ourselves to go into phase 3. And this phase 3 was also, as you can imagine, was also very good. We had very good results, because we demonstrated that we have statistically significant results that the pain intensity with our compound over 48 hours was better than tramadol, hydrochloride, celecoxib, and placebo. And this can be seen in this picture, where Seglentis, you can see that this is the pain intensity difference from baseline to 48 hours after a postoperative bunionectomy with osteotomy pain. Then you can see here the placebo, what the patients felt. And here you have the tramadol hydrochloride, and here is the lecoxib. And our co-crystal was able to get a better response. So that's why the authorities, mainly FDA authorities, consider that co-crystal has to be, the co-crystal Seglentis had to be approved, and it was approved in October 2021. So imagine that even it's, let's say, a drug that was already known. We started in 2008, we end up in 2021. So it's a long time for the phase 3 studies. And as I told you before, we were the first, or we believe that we were the first, but in 2015, Entresto, which is a co-crystal of two APIs, valsartan and sacubitril, was launched to the market by Novartis. I happen to know this a little bit before because a person who was advising Novartis, a crystallographer that was advising Novartis, was in a meeting, and by chance he explained the story, and then I realized that our co-crystal will not be the first. But nevertheless, it's always good that such a big company as Novartis gives you the guideline to do it, and that this can be done. Other big companies like Merck Sharp and Dohme or Astellas are also pursuing, also launched co-crystals to the market, but those co-crystals are not API, are co-crystals of one API with a coformer, okay? So far, only two API co-crystals are in the market. One is from Novartis for cardiopathy and hypertension, and this is from Esteve for treating analgesia. Just to say that Esteve arrived to a license agreement with Kowa Pharmaceuticals in America, and Kowa will be the one selling the compound in the US. So this is the end of my talk. Just a small detail that Esteve, after so many years and after having this success, decided to transfer the preclinical research to WILAB, which is a technological center for non-profit organization. So we moved, we didn't move, by the way, from the site, but we moved from the company. Instead of Esteve, we were working for WILAB. And at the same time, I'm working also as a senior vice president in medicinal chemistry here at ABAC Therapeutics, also in Barcelona. And my two messages here will be the same of Professor Suma. Don't just work to get a big profit. Profit will come by itself. And the other one is always find some innovation, always find opportunities, because a small innovation can give you a big success. And I am happy for this, and also for the company who invest a lot of money during many years. So thank you very much.
Thank you, Tony, for this further interesting story. And I also appreciate the second message, because yes, maybe you don't need to make a million of compounds to get the right one, but simple, brilliant ideas, always from the mind of medicinal chemists, can be very successful and can help patients. So thank you also for highlighting this. So now the talks have ended because unfortunately Professor Rui Moreira, the president of EFMC, couldn't participate. And so what we are have planned now is to go for a second round of round-table discussion. So in the remaining time, I would like to ask our speakers, because we have the chance to interact with them, some questions, but I'm starting, but then I'm waiting for your questions, okay, for you, because maybe you have something that you want to know regarding these interesting stories. So maybe I want to start with Vincenzo, because if you have understood. To me, it's very interesting to know how transition in career should be phased and how they change your life, your work, and even the daily activities and the motivation. Okay, so a little bit more about your this change.
So, as was mentioned, I always said that I have now 3 lives. So the first one was in Merck, working for a really big pharma, where I discovered Isentress, but also another drug for HCV. The bad shock, working for big pharma is you have everything you can think about in terms of resources, money, and you can do everything you want. So it's up to you. And it was very nice, a competitive environment with the US people, and it was fantastic. And then I discovered what does mean working for big pharma. From one day to another, they announced the closure of our research site, in spite of the fact that we put on the market 3 different drugs. So we rescue that started my second life. So we tried to rescue our research center, and so we did this together with other director. We reopened IRBM, and we started as a CRO, so contract research organization. So we have been working for making science for other people. It was very tough. I was vice president there, and 10 years, the company is very healthy. IRBM, the actual IRBM is very healthy, very nice. Like the Schindler List, I re-employ or the majority of my former colleagues. And then I say, okay, now I've done also this. So as like an entrepreneur, I rescued the company. It's healthy, it's going. So I want to do something different. And it was big change again. So I became a professor, a completely different world. I think my previous 2 lives can really help you in give you the idea that if you pursue something that you strongly believe in, what is your passion for science, whatever it is, if you follow that passion, you can have very nice and good results. And so my vision now is that I should have been able to transfer to you this vision that it's up to you what you can do. And especially, with the students, there are some students here, my students, and I believe that they really trust the fact that academia is one part of the life, but also the industry is very important. 2 different aspects, 2 different stories, and I cannot say one is better than the other, are different. We do different job in the 2 environments, but they are interconnected, because one without the other doesn't make any sense to me.
Thank you. I consider these words particularly precious for our society. Okay, and so are there questions from you? Cyril. Cyril, could you come here, because we are connected on YouTube, so you should speak on this microphone.
Thank you very much for this opportunity, and thank you very much for giving me the mic to ask the questions. I can see very talented people here and very inspirational. And my question is, how can you see the future now of drug discovery, and how do you see it? Very bright, or is there any difficulties we have to face on?
This is for
everyone.
For everybody.
Everyone, yeah.
So from my perspective,
Maybe because this is a very important question, maybe we can be a quick answer for everyone. So okay, so you can even listen to the different perspective. What do you think?
Yes.
Okay. So please.
So, yeah, in my opinion, drug discovery is changing. So we have several new technology that help the drug discovery. And so it's a really, it's a changing field. It's very attractive, very interesting. And I don't think there is any point in thinking there is no future for the drug discovery. Absolutely. So the pandemic is a lesson for everybody to me.
Maybe Tony?
Yeah, I agree completely. The future of drug discovery, I think it's bright. Also the small molecules, because a lot of people think that only biologics will be from now on the only drugs approved. But we will hear Professor Tiziano Bandera the last day of this school, and he will explain all the new compounds that have been launched to the market. I think that the landscape has changed mainly because big pharmas are less and less taking the risk at the beginning of the process. So what I explained here in my particular case, that we were from the beginning to the end doing all the process, and now is less and less happening. So big pharmas tend to let the risky part of early research on biotechs, startups, but this means also that a lot of people has to be allocated in these startups, and even CROs, like Tilly said, CROs are also contributing to this academic environment, to this scientific environment. So probably people will not be in big pharmas like we probably been, but more and more they will doing new modalities and risky things, more innovative things in the small biotechs, most likely. And this, but the future for drug discovery, my only concern probably is that less people wanted to do drug discovery, because I don't know how many of you here that are studying medicinal chemistry would like to do drug discovery. Can you raise your hands?
It's quite a lot.
It's quite good, because I made the same survey in other forums, and I can tell you that less than the 50% of medicinal chemistry students want to do drug discovery. They prefer to enroll in other disciplines or more project management or whatever. But
Thank you. Monica?
Yes, from our perspective, I think, well, I agree totally with what Anthony said. And in particular, if you think, for example, to our company, the business model we have adopted really is to start from the preclinical and translational science and leaving the drug discovery part to small biotech, academia, so where you have really apart from the risky, because risky from I don't know which kind of perspective, but really you have the science for each protein, mechanism of action, and you can build really a good drug that then can be developed and brought to the registration by the company. So I think, at least from us, from our perspective and our, business model, is something that the Italian university should really implement at the best, because it's really important.
Thank you also for this last message. Tilly, we want to know even your opinion.
Yeah. Oh, I'm over here.
Yes.
Can you hear me? Can you hear me? Okay.
Yes.
Yeah. So I think the future of drug discovery is very bright. I think biologics and new modalities is very strong, and there's a very positive push in that direction in drug discovery, but they do tend to be expensive. And another big trend globally in the world is to look at cost of our healthcare going forwards. And small molecules will always be the cheapest option. Yeah. So you begin to see that now with programs where maybe the first drug for the novel target is an antibody, but then there is a strong push. They maybe take IL-17 or some of these drugs. Then there's a strong push to try to deliver a small molecule alternative because it will deliver the long-term healthcare needs globally, which are cheaper medicines. So yeah, I think the future is very strong. Yeah.
Yeah, I personally agree 100% with this concept. Especially, yeah, we already mentioned the problem of access of drugs to medicine. So what you said is very important. Thank you. And thank you for this very, very interesting question. Thank you. Okay.
Thank you very much for answering.
Okay, thanks. So, any further question for you? Please, you should come here, always for the problem of the microphone. I don't remember your name. Eduardo, please.
Thank you very much for your presentations. And so, okay, the future may be bright,
Yes.
but
everyone is convinced.
yes,
no?
or I hope so. But do you think
Right.
that artificial intelligence will replace us?
Wow.
Like, do you think that artificial intelligence may replace us in the future, particularly in retrosynthetic planning or or synthesis in whatsoever?
Again.
Yeah.
Do you want to start? Okay, yes, Monica, please start.
So I think it will be of help for sure. So there are already companies that are applying the drug discovery part, starting from the artificial intelligence, that are really going very fast. And we have really many examples. But still, I think the curiosity and the passion cannot be for sure substituted by a machine. So I think it will be of help in speeding up the development. This is something that all of us, we would like to see because, as we mentioned before, sometimes took at least 10 years, 15 years, and the patients are in need. So having something that can speed up some very boring processes, it will be useful, but I think no one can really substitute the passion, the curiosity, the commitment of a human being. So use it in the proper way. This is my message.
Thank you.
Tilly?
Yeah, I think AI will be really helpful and already is very helpful. And the area where it can have the most impact is in selecting the right patient populations who will have responses. And we can also be using that information of understanding disease better at a genetic level to design better programs. So I think it will have a positive impact on drug discovery, and that's the most immediate area where we can see the benefit. But it will mean we can do things faster. I don't think it will mean you don't need humans. Humans are very adaptable. We will adapt and use AI to our benefit, I think.
Thank you. Tony?
Yeah, once I heard a sentence that I like very much. It says something like, artificial intelligence won't replace any chemist, but the chemists who don't apply artificial intelligence will be removed. And this is, I think, the mentality. Artificial intelligence and the new modalities are what we saw today of virtual reality are not coming to expel us from the work, but to help us. And then I think that mainly young people like you should be like we were in the computers, When I was very young, there was no computers. So we need to use the computers, and the same question arose at that time. These computers will make us you remember. That's the same with the electric guitar and the electronic music. So it's always those things that are coming that, in a way, we are afraid of. But I think that artificial intelligence, like computational chemistry, virtual reality, comes to help us. So I'm pretty convinced on that. No one will replace. Moreover, I think that there will be few medicinal chemistry for what we will need in the future. And we need more students and more people involved in medicinal chemistry, not less.
Thanks, Vincenzo.
So I totally agree. So will be another tool that we can use to make easier our life and maybe better drug. You know, in the medicinal chemistry history, you know, for example, combinatorial chemistry was one of the revolution, and people say we don't need to make a single molecule, we will always test a library of compounds. The reality was not true. Same for artificial intelligence. We will use the best part of the artificial intelligence as a tool that we can use.
Thank you very much.
No, thank you, Eduardo. Another very interesting question, so
Yes.
thank you.
And I hope you're right.
So there is time for the very last question. No? Yes. Please. What's your name? Davide? David. Ah, David. Where are you from?
Spain.
Spain. Please, David.
Thank you for all the presentation. Has been great. And I always had a question about academia and then working in industry. How do you think it's affecting? Because, as you said previously, I think academia and obviously industry, they must work together. How do you see the future in that sense, in terms of, for example, identification of new targets, mechanism of the reactions, that collaboration between both? Do you think it's going to be stronger during the next few years, or that's my question?
Thank you. So, Vincenzo, it's your turn.
Clearly, you know, there are, I think, you are now in your training period, and so you know the university world, no? And there is the feeling that in the pharma there is not real research or a deep understanding of new mechanism or new reaction, whatever it is. In the reality, it's not true. So I just mentioned a few articles that have been published on Science that was, you know, breakthrough in the understanding of the mechanism of the inhibitor, the mechanism of the integrase. So the girl there, Daria Zuda, is the person that was able to put in the test tube the HIV integrase enzymatic cassette. It took 10 years. So everybody tried and nobody succeeded. So there are, in the university, I think the most important mission is really to form very solid scientists with a clear vision what they are doing. So you have to be an expert in what you are studying. You must get a passion on your topic, know everything, learn how to do a good experiment, and really get in love with what you are doing. Then you can decide if you want to really investigate I would say the basic science, that is your place. If you want to go in a more practical aspect, so, but looking always the science, you move. So in I never thought, I did my PhD, but I never thought to be in the university, never. So just now that I have white hair, I decided to go back to the university. And simultaneously, the company looked what the university is doing. So there is really, as I said, a cross fertilization between the two aspects. I thought that now what you know is just one part, but try to really understand what the other part is doing. And, you know, going back to the industry, they should tell us, because now I'm in the university, tell us what they want, what they really need from the university. I think this is, in the US it's much more clear, in Europe much less. Because there are, you know, the university is like a closed environment and the industry is another closed environment. But now they are getting close together because we need the cross-fertilization.
Tony?
Yes, I'm very optimistic on that. It's a very difficult question, by the way, because I remember that 30 years ago we were already discussing about this, how to bring together industry and academia. I think that there are very positive things nowadays. For instance, the startups. I think that the startups from the university is a good way that the people from the academia understands industry, because as he said, we are different worlds, but don't forget that all the people that is in the industry has been at the university first. So we know better both worlds, probably. Then the startups will help the people at the academia to have interactions with industry and so on. And to me, the big barrier that separates us is probably the confidentiality issues, because we have different timelines, and confidentiality is very important for the company. And then for a PhD student, what is very important is to publish results, and to publish quick and fast. And so people who is doing the PhD in collaboration with the industry has a kind of handicap in this, because they cannot publish as fast as they want. And at the same time, the industry is the same. We are forced to publish as soon as possible, and sometimes we need to wait to have the patent on the right time. And this is, to me, in my opinion, something that is a barrier that separates us. But I guess that sooner or later this will be overcome.
Tilly, I know that this is a topic you are very fond of, so...
Yeah, no, I think it is a challenge at the moment, and at the root of that challenge has just been articulated. It's that industry precisely does not want to tell people what it's doing, because it wants to keep that secret for as long as possible. I'm not saying that's a good thing, I'm just saying that's how it is at the moment. And academia actually wants the opposite, where possible, to publish that information, you know, as soon as you can, to be the first to disclose that discovery. And that is the point of motivation that's different in that area is what causes most of the issues, in my experience. Yeah.
Thank you. And Monica, just to
yes,
sum up.
I think most has been already said, so I think really that the long term, the mission is completely different, of course, but still, I think in other countries, this dichotomy in some way has been solved. So in particular, if you think in US, where a lot of cross-fertilization between industry and academia is in place. We all know that there is a problem of IP, confidentiality, different timelines, but still I think we need to make an effort, because for sure it's true that also in company we do science, we do research, but still, and if I can make an example in oncology, now you don't have a single disease, no, there are subtypes of any single, in lung cancer you have now, I don't know how many different subtypes, the same in all the other diseases. So you cannot be expert of everything. So going to the university where no, the idea, the protein was cloned, I think it can make the difference. So we need to find a way how to collaborate, recognizing the different mission and the different patent protection issue or everything. But I think looking at what has been already solved, we can really apply this also in Italy.
Thank you.
Thank you. So unfortunately, time is over, so we should close. I personally enjoyed a lot the round table, so I truly thank all the speakers. Just to wrap up, few words by me. So what we have learned today is that drug discovery is complex, but we can make it. The future is bright, but we need collaboration. we should strive for collaboration for the benefit of the patients. So thank you very much for participating.